nice for me to be here. It's my first time in Colombia, so I'm very excited to be here. And let me start by introducing myself. I work as an assistant professor at in the Genetics and Aging Research Unit at the Mass General Hospital in Boston. I did my graduate studies back at the Health Policy Research Center in Munich. And after that, I did my postdoctoral training in the lab of Dr. Rudy Tanzi at the Mass General Hospital. And for many years, I've been investigating the function of the innate immune receptors, CD33 and TREM2, and how mutations in these genes contribute to Alzheimer's disease pathogenesis. And during the past 5 years, we've been implementing also gene therapy studies for neuroinflammation based on targeting CD33. And over the years, I've received several awards, including the NIH Pathway Independence Award and the Innovation Award from Mass General Brigham. And these types of awards have facilitated my research on a daily basis. So today I'll be presenting my research application for the CARD Fund, which I'm very grateful for. And I'll start by saying that in Alzheimer's disease, there is accumulation of extracellular amyloid beta plaques. And in the brain, there is a cell population that we call microglial cells. They represent about 5 to 10% of the total cell population in the brain. And they're considered to be the sentinels of the brain with a very important function to participate in the clearance of amyloid beta and also cellular debris. Thank you. To participate in the clearance of can I go on now?
Yeah. Fine.
Okay, is that okay?
Yeah.
So the microglial cells are the sentinels. Do you want me to switch it off and just speak very loud? Yes. So the microglial cells are the sentinels of the brain. They're very important for the clearance of amyloid beta over here and cellular debris. They can also secrete pro-inflammatory mediators and have a less beneficial function where they become pro-inflammatory and they enter a vicious cycle. It is known that impaired microglial phagocytosis can enhance amyloid beta accumulation, which ultimately leads to more neuroinflammation. And new evidence shows that reduced neuroinflammatory response may lead to human resilience to Alzheimer's disease, even though there is presence of plaques in neurofibrillary tangles. And just to summarize,
We have a technician here who's going to try to adjust this microphone. So we need to turn it on so he can hear the feedback. Keep talking.
So the microglial cells, as you can see over here in their homeostatic state, they have a very important function where they participate in clearance of amyloid beta and cellular debris. They can also contribute to excessive pruning of synapses. Synapses are very important for cognitive function in the brain. However, they can also become pro-inflammatory activated and contribute to inflammation in the brain. So they have a dual function, and there's a lot of research focusing on understanding microglial cell function in the brain. And my work today has been focusing on investigating the role of the innate immune receptor CD33 that is expressed in these microglial cells in the brain. And CD33 has been identified as a risk factor for late onset Alzheimer's disease, first time by Dr. Rudy Tanzi's lab back in 2008. And after that, several studies have published in Nature Genetics that the minor T allele of the 4 single nucleotide polymorphism in CD33 is also associated with late onset Alzheimer's disease. Now, as I said, it is an innate immune receptor that is expressed on the cell surface of the microglial cells. It has a ligand binding domain, the VIG domain that I'll be talking about today. It also has a signaling platform that we that is constituted of two domains, and we also call it the inhibitory tyrosine inhibitor motif over here. And CD33 is activated by sialic acids, and it regulates inflammatory responses in the brain. It is also expressed in monocytes in the peripheral system, and therefore it has very important functions. Okay, so here I tried to summarize in a visual our research that we did some years ago using post-mortem human brain samples from AD patients. And basically what we found back then by looking at the post-mortem human brain samples is that AD patients that carry the common variant of the 4,4 single nucleotide polymorphism, they show high CD33 expression levels in the brain. They also show high amyloid burden, which is due to the limited degradative capacity of these microglial cells highlighted in red. On the other hand, carriers of the protective variant of the protective allele of the 4,4 signal nucleotide polymorphism in CD33, they show low CD33 expression levels, low amyloid burden in the brain, and enhanced degradative capacity of the microglial cells highlighted in red. So that was the first indication that showed us that it is actually good to have low CD33 expression in the brain, we decided to investigate at the molecular level what CD33 does in microglial cells, and also later on try to understand how we can inhibit CD33. So that research with human brains was very, very important to us. And as I mentioned, we later on designed experiments at the molecular level in microglial cell cultures by using different constructs of CD33. And basically CD33 generates two splice variants of full-length CD33 that you can see over here. And it is produced by the AD risk allele that we just spoke about earlier. And it is expressed on the cell surface, and it leads to inhibition of amyloid beta uptake in the cells. It also generates the shorter CD33 delta VAG isoform over here that lacks the ligand binding domain produced by and it is produced by the AD protective allele. And as you can see here, there is no ligand binding domain, and we know that the ligand binding domain is encoded by exon 2 in CD33. And I'll be talking a little bit more about that in the next slides. And basically our research showed that when CD33 was under this short isoform in cells, it did not inhibit microglial clearance. So that showed to us that the ligand binding domain of CD33 is required for the CD33 mediated inhibition of microglial uptake and clearance in cells. So basically we really needed this domain to be present to see the detrimental effects. Whereas if the ligand binding domain is not there, there is no more detrimental effects. And actually this isoform turned out to be beneficial as later research by other labs has shown. So other labs that have been working in parallel on CD33, they reproduced our research showing that full-length CD33 is expressed on the cell surface of the microglial cells, and it inhibits phagocytosis. They also found that it inhibits proliferation and migration. That is important for microglial cells because microglial cells, when they sense a threat in the brain, they tend to migrate to that, or they, migrate also and are chemo-attracted to amyloid plaques, and they try to clear them. And it looks like CD33 inhibits all these beneficial processes. So it is at the cell surface and full life. On the other hand, the short isoform of CD33 that lacks the ligand binding domain has been shown to be expressed inside the cells, probably in peroxisomes. And it actually does the opposite, generates the opposite effects of the full-length CD33. So it leads to increased phagocytosis in microglial cells, and it induces proliferation and migration, which is actually very, very good. for the brain function. So, years ago, we asked ourselves, does inhibiting CD33 represent a potential therapy for Alzheimer's disease, and how can we do this? And that's how one of our earlier projects started. And before I go on, I would like to specify that a lot of our research at Mass General and also in other labs set out the platform for biotech and pharma to select CD33 as one of their major drug targets for the treatment of Alzheimer's disease. And basically, there's been two companies that have been trying to develop CD33 specific monoclonal antibodies so that they can apply immunotherapy in Alzheimer's disease. One of the antibodies that has been called AL003 has been developed by Alector in collaboration with AbbVie. And although it had promising effects and it has proven to be safe and tolerable in phase 1A study, at low doses. Unfortunately, it had side effects at higher doses, and therefore the clinical trial has been terminated, unfortunately. And we think that the trial may have been terminated due to the on-target toxicity following CD33 inhibition in the peripheral immune system, because the antibody was systemically administered, and a couple of percent of that antibody go into the brain. However, some of it had on-target toxicity probably on the monocytes in the peripheral system in the blood. And there's another company that is called Alchemab Therapeutics. It's a mid-sized biotech. And that's the very interesting part, and it highlights again how important CD33 is for Alzheimer's disease. What they did is they sequenced the cells from the immune system from individuals resilient to Alzheimer's disease. And they found naturally occurring CD33 antibodies. So people who did not develop Alzheimer's disease, they had antibodies that were naturally targeting CD33. It is unknown how they enter into the brain as of now, but those researchers identified one of those naturally occurring antibodies, and they found that it engages with the ligand binding domain of CD33. So instead of reducing CD33 levels, it acts as an inhibitor of CD33. And we think that this antibody may be promising for therapy. However, it has not entered into clinical trials yet, and they will be trying to target microglia in the brain, but again, the clinical trials have not started yet. Meanwhile, on our end, we've been trying to inhibit CD33 by using a gene therapy approach. And I would like to emphasize that microglial cells, they're very difficult to transduce with adeno-associated viruses that I'll be calling them AAVs. AAVs are a great tool to transduce astrocytes, neurons, but very difficult to transduce microglia. And for this project where we've been trying to inhibit CD33 function using gene therapy, I've been collaborating with Dr. Casey McGuire at the Mass General Hospital, who has been developing a lot of AAV tools. And basically what we decided back in the time was to transduce astrocytes in the brain with vectors that encode an artificial microRNA targeting CD33. Once it binds to the transcript of CD33 in the microglial cells in the brain, it will lead to its degradation. So we did an indirect approach for transduction because these vectors transduce astrocytes first because microglia are so difficult to transduce. And then the microRNAs targeting CD33 were exported through extracellular vesicles from astrocytes into microglia, and here they led to reduction of CD33 levels. So it was. Yes.
Do me a favor. The crowd. Can you either use another word or explain transduced?
Yes, absolutely. So basically, these vectors, once we inject them, for example, in animal models, we want them to go in the brain and then inside the cells. When we use AAVs, we hope that they will transduce the cells. It's basically the process of entering inside the cell. Again, these microglial cells, they're very, very difficult to be infected or transduced with these AAV vectors. We still pursue this approach while waiting to develop better tools. And as I mentioned, we have here the microRNA targeting CD33 inside the vector. And by using cellular models, we have shown that the microRNA targeting CD33, it leads to about 35 to 40% reduction in CD33 expression levels. And we used that vector to inject our mice in the brain. And in this case, we used mouse models of Alzheimer's disease. And by using the GFP over here, we have shown that by using microscopy that these vectors are expressed in the mouse brain. They do express also the microRNA targeting CD33. And I'll try to simplify this, but we've started analyzing the mice that were injected with these vectors, and we tried to understand whether this method is efficient and whether we can reduce CD33 levels indeed. And we looked at the brain. We looked at the CD33 expression levels in the brain tissue, and we saw again about 35% reduction of CD33. And we also looked at the amyloid beta pathology in the brain lysins, and we saw that this approach helped us reduce the soluble forms of A beta 40, and a significant reduction, a trend in reduction in the soluble A beta 42. We've also looked at the insoluble A beta, which is over here, and we saw transfer reduction. After that, we went on to look at the amyloid beta plaque burden in the brains of the mouse models that were injected with the AAV vectors containing the microRNA targeting CD33. And we saw that when we quantified the amyloid beta plaque load, which is highlighted here in red, we saw that we reduced the amyloid beta plaque burden by about between 30 and 40%, both in the cortex and hippocampus. We also looked at the number of plaques. We call it also density, and we saw a good trend in reduction. And I would like to emphasize that here we used an early intervention approach where we injected the mice at about 2 months old, and we sacrificed them later at about 8 months old, and we saw these lasting effects. And these AAV vectors, they've been in the brain for a long time, and we've used only one single injection. And we've also looked at the effects of the microRNA targeting CD33 in the AAV vectors on the expression levels of pro-inflammatory cytokines and chemokines. So these are pro-inflammatory mediators that are secreted by microglial cells. And chemokines are also important for the migration of microglial cells. And we saw that when we inhibit CD33 function, we found that the pro-inflammatory TNF-alpha cytokine expression levels were decreased. We also found a good trend in reduction for the expression levels of the CCL2 and CXCL1, both of which are chemokines. And so basically, this approach showed us that by targeting CD33 through gene therapy methods, we can lead to reduced A beta accumulation and reduced neuroinflammation in mouse models of AD. And moving forward, while we were happy about these results, we kept on thinking how we can develop better tools so we can target human CD33 instead of mouse CD33, because there is some differences between mouse CD33 and human CD33. And so we wanted to develop models that would move us closer to the clinical side of research. And so what we did is we generated a humanized CD33 model. And basically here you can see that we still kept the endogenous mouse CD33 promoter, but we used the sequence from human CD33 and we replaced mouse CD33 sequence with the human CD33 genomic sequence. So this is a humanized CD33 mouse model that we then crossed to the 5xFAD mouse model of AD. Because the humanized CD33 mouse model of AD, the humanized CD33 mouse model, it does not show a beta pathology on its own. So we basically crossed the humanized CD33 mouse model with a mouse model of AD that we call 5xFAD, and that expresses two transgene, APP and PS1, that will help the mice to develop a beta accumulation and neuroinflammation. So we generated this compound mouse model of Alzheimer's disease, 5xFAD human CD33, and we started by characterizing it before starting to use it for gene therapy or other approaches. And as you can see here, we've shown that human CD33 is indeed expressed in the brain, and that this approach of replacing the mouse gene with human CD33 has worked. We've also did stainings for the microglial marker IBO1, and we also stained for A beta, which is in the blue channel. And basically here you can see that IBO1 positive microglial cells, they express CD33 and they cluster around plaques over here. And basically this humanized mouse model will be used for future projects, including for the project that is about to get started with the CARD fund. And we'll be using it for therapeutic strategies to reduce CD33 expression, modulate CD33 splicing, and also to inhibit CD33 function. And basically, I have a couple of slides just to show you what we will be doing in this specific project that we're very grateful for to the CARD fund. We're trying to develop novel tools to inhibit CD33 in the brain that will be more efficient. And we decided to use antisense oligonucleotides that I'll be calling ASOs. They are short, single-stranded synthetic RNA or DNA that binds to the transcript of CD33 or another gene in a complementary manner, and it will lead to its degradation. This is one method to use ASOs. There is some ASOs that are used in other neurodegenerative diseases and neuromuscular diseases, and they're injected intrathecally in the cerebrospinal fluid, and they have been shown to distribute broadly in the central nervous system, and they localize across various cell types, including microglia. And that is very important to us because it is so difficult to enter with tools into microglia to inhibit CD33 function. And basically here, we propose targeting CD33 by using antisense oligonucleotides in our humanized 5xFAD human CD33 mouse model. And basically here, as I said, ASOs, they bind in a complementary manner to the mRNA or to the transcript of the gene of interest, and they lead to its degradation through the RNase H1 mediated degradation, or they can also prevent protein translation. So that's the next level once the transcript will generate a protein. However, ASOs can be used also for splicing modulation that we would like to also pursue in this project, and I have here something to show you about the splicing. As previously mentioned, CD33 generates two splice forms, the long full-length form that is expressed at the cell surface. It contains all the exons, including the exon 2, and it is generated by the RISC AD allele, while the short isoform that lacks exon 2 and lacks, therefore, the ligand binding domain of CD33 is beneficial in the brain, and it is generated by the protective allele of the 4,4 single nucleotide polymorphism. So that happens in the brain, and we call it an endogenous splicing. However, there is another group that used this splicing mechanism to generate a splicing reporter. And they generated a splicing reporter because they wanted to find compounds and ASOs to target CD33 and to modulate splicing. And they, by using this reporter that encodes luciferase in the exon 3, they found the ASO 79, so it's an antisense nucleotide 79 that allows to exclude exon 2 and therefore generate the short isoform. And this is one of my last slides where we show the aims for the project. So first of all, we'll start by screening ASO sequences in a microglial cell line over here that expresses human CD33. And then we will validate it in the humanized CD33 mouse model. And we will be first looking at the reduced expression levels of CD33, basically ASOs that lead to transcript increased levels. And then we will take the best ASOs to that mediate CD33 suppression and test them in the compound 5-XFAD humanized CD33 mouse model and looked at all the effects at the data accumulation and neuroinflammation level. And we also plan to test the ASO 79 that has been shown to modulate 33 splicing and look at the CD33 isoforms, A-beta pathology, neuroinflammation, and also cognitive function. We have a behavior facility for animals at the Mass General, and we're very eager to see whether cognitive function will be improved by using these ASOs. And basically our long-term goals is to test efficacy and safety of CD33 targeting ASOs for future therapeutic applications, to provide a proof of concept study that ASOs targeting CD33 can reduce both accumulation and A-beta accumulation and neuroinflammation, as well as improve cognitive function. And finally, we hope that these studies, by looking at the microglial phenotype and the cell population targeted by ASOs in the brain, will help us better understand the mechanisms that control microglial function in Alzheimer's disease. And I'd like to thank the Genetics and Aging Research Unit Director, Dr. Rudy Tanzi, for outstanding mentorship, scientific guidance, and help through all this work and this project, and the talented team of scientists that I've been working with, my collaborators, Dr. Casey McGuire and Eloise Udry, and all the funding agencies and the CART Fund for funding this project. And I'd be happy to take any questions. Thank you.